Welcome to Barry's In the Interim podcast, where we explore the cutting edge of innovative clinical trial design for the pharmaceutical and medical industries, and so much more. Let's dive in.
Well, welcome everybody back to In the Interim. I'm your host, Scott Barry. And for today's topic, I feel a little bit like years ago, I was on faculty at Texas A&M. I was assistant professor of statistics at Texas A&M. And just starting at A&M, I had come from graduate school, came from Carnegie Mellon. And I don't know if they still do it, but Texas A&M had, at the time, a policy that every graduate student, everybody pursuing a dissertation, would get a random faculty member on their dissertation committee. So if you were a statistics graduate student doing a dissertation, you might get a faculty member from meat sciences or from something to be on your dissertation committee, a voting member of the dissertation committee. And so when I got there, I was assigned to a students committee. And it was, when I walked in, it was sort of like, oh no, the generally throughout all of the graduate programs, the worst thing that could happen to you was getting a statistician randomly assigned to your dissertation committee. So I think the first one I did was forestry and crop sciences, and they're actually doing experiments. It was fascinating. And the second one I think was wildlife and fisheries science. It was a fascinating project where they were following wolves and they were tracking them and they were reintroducing wolves into the wild and they were trying to figure out their behaviors. It was dynamic data. It was absolutely fascinating, but they were fearful of a statistician walking in like, oh no, I now have a statistician on my committee. Now, what does that have to do with today's topic? Today's topic, I don't think it's the same sort of scenario because statisticians are rampant in the clinical trial world. But I am going to talk about today a look at a recent issue of JAMA. So a wonderful thing about clinical trial design is you can go see the results, the published results of other trials and see those trials design, see how they carry out the analyses, the interpretation of those. And of course, JAMA, New England Journal, Lancet, all of these provide this really nice opportunity. So I make sure whenever possible to open up these trials, to check out the design, see what they say, what's being done in the world of trial design. What would I have done differently in them? And do I agree with the interpretation from the medical journal? So I get to jump in a statistician review, and I'm going to talk about them on this show. So I've done one other. You can go back to the episode where I sort of randomly fell upon a really, really interesting trial of using iron supplements in heart failure where the patients are deficient in iron. Fascinating trial. So you should go back to that episode. These trials, I'm going to look at two trials the email comes in from JAMA and I click on them. And, these are interesting enough. I want to talk about these. Okay. So a little bit is doing this a bit publicly, and I hope people don't say, oh no, a statistician is reviewing my trials in this setting. By the way, an interesting thing, one of the more interesting things I got to do when I was at Texas A&M, I was a columnist for Chance magazine. It's a publication of the ASA. And each quarter I got to write an article that was a statistician reads the sports pages. So I got to go in and find topics that were coming out. And I did this for 10 years. It was a quarterly magazine. So I wrote 40 articles about reading the sports pages, things that come up in sports, statistician's interpretation of them. So this is a little bit like that. This is a statistician reads JAMA, and you know what do I look at when I read these trials? Okay, probably a bit of a bias in the trials that I look at, but this is a recent episode of what showed up in my mail. So the first trial was published June 4th, 2026, and it's called the TRACK, Randomized Clinical Trial. It is a trial of, and the title of it is Low-Dose Rivaroxaban in Cardiovascular Events in Advanced Kidney Disease. So a little bit about the trial, and I am not a clinician, so I'm reading the article of this. So rivaroxaban is a anticoagulant, and apparently anticoagulations in some form of kidney disease with aspirin have demonstrated good clinical outcomes. So this is a trial looking at low-dose rivaroxaban looking at, does it alter cardiovascular outcomes for patients with advanced chronic kidney disease? Now, these are CKD, chronic kidney disease, stage 4 or 5, including patients on dialysis, and they're randomized to a rivaroxaban, low dose or not, in the trial. And the primary outcome is a composite of cardiovascular death, non-fatal MI, stroke, or peripheral artery disease event. And the primary safety outcome is major bleeding, which is typical of trials of anticoagulation. The risk, of course, are bleeding events in that. So that's the trial, the major part of what the trial is trying to address. Now, a little bit about the trial design. It's an event-driven trial. It's a little bit, I found it a little bit confusing in the trial design. It's an event-driven trial, but it talks about the trial is designed to enroll 1,900 participants over 3 years with an expected follow-up of 5 years. So the 1,900 patients would provide statistical power to detect a reduction, so a hazard ratio of 0.75, would have 90% power to detect a difference. They're going to do a log rank test. They're going to look at the hazard ratio in a Cox model for the cardiovascular events, time to first event. Now it does, and then it says the 1,900 is a bit, they're going to enroll that, but they're going to wait for the events. It's an event-driven trial, 550 primary outcome events, assuming a log rank test. 90% power for a 0.75 hazard ratio. And it also says the number of events provides 80% power for a 22% risk reduction, 0.78. We'll come back to that interesting 0.78 has 80% power in the trial. Now, the first thing I tried to diagnose from the trial was this adaptive in any way in the trial, or is it a trial and we're going to wait 5 years and look at the data? Well, it says 2 formal interim analyses are planned to assess efficacy when approximately 1/3, which is pretty early in event-driven trials, and 2/3 of the primary endpoint outcomes have been observed. They use Hay-Biddle-Peto stopping rules. It says using 3 standard deviations at either of the first 2 interims. So that's a nominal alpha of 0.27 within that. So the typical 2.5. This is 0.27. which it describes minimal impact on the nominal type 1 error rate by the end of the trial. So 4.82% is the alpha. So at the end of the trial. So it preserves a good bit of it. Now, it says nothing about futility analyses in here. It says a detailed DSMB charter will be developed before starting the study in consultation with DSMB members. That's essentially the extent of it. I looked through the SAP, which was an appendix to this, a supplement to it. Looked through the SAP, and this is really the wording it gives there. I think in hindsight, the answer is there were no futility analyses in the trial. So it's a superiority trial for rivaroxaban on cardiovascular events. Now, what happened in the trial? So it says, and this is in the paper, in the report, on July 8th, 2025, during a pre-specified review of interim data, and it's kind of interesting because it says 254 primary outcome events, which is 50% of the planned events. 1,432 patients randomized. Remember, they said 1,900, so 1,432 randomized. And it's at 254 events. They said that they're going to do a superiority at 1/3 of events and 2/3 of events. So it says on July 8th, 50% of the events, 254. The Data and Safety Monitoring Board recommended early termination of the trial to concerns regarding net harm and a low probability of demonstrating efficacy. Okay, so the DSMB made this recommendation, and then it says the recommendation was not based on pre-specified stopping rules. So from all my read, there were no pre-specified futility, but the DSMB makes this recommendation. Net harm and low probability of demonstrating efficacy. And then it says, it gives additional detail. It says, rather, the decision was based on a post hoc analysis estimating a conditional power of 16% for a hazard ratio of 0.78. Indicating a low likelihood of demonstrating efficacy even if the trial had continued until the planned 515 primary outcome events. Okay, so a lot to unpack here. So no pre-specified rules. They do a post hoc. I think with no pre-specified rules, everything is post hoc. So I found that wording somewhat weird. Within that setting, as though that's super relevant. And they do conditional power, and they say a low likelihood of demonstrating efficacy. So first of all, what is conditional power? 16% probability of demonstrating superiority at the final analysis, and it says 16%. Now, conditional power is given the current data at the interim at 254 events. It's a calculation of what's the probability of seeing superiority at the end of the trial. Now, conditional power assumes a single value for the hazard ratio, and it can be done in many ways. Sometimes conditional power looks at the current estimate of the hazard ratio. Sometimes it uses a preset hazard ratio, like it did here. So it assumes a hazard ratio of 0.78 for the rest of the trial, with variability of the rest of the data. Suppose the truth is 0.78. What's the probability that at 515 events, we see superiority? That's the calculation that's made. And that's 16%. My first reaction is that's not that low. That's that likelihood, I don't find that low at all. it was interesting. My son, Cooper, who I've talked about on this show before, he was in 7th grade, and we would not let him play tackle football. And he came to us with an argument that he should be allowed to play tackle football. And as part of his argument, he found data that said the likelihood of a neurological event for a kid playing tackle football, a serious injury, was only 13%. And he presented that in his PowerPoint presentation to us. Yes, we have a weird family in this, which that was only 13%, so he should be allowed to play tackle football. And our reaction was, oh my God, 13% is enormous. The 13% chance of serious injury in that setting is very large. So 16% probability of demonstrating superiority by the end of the trial doesn't strike me as that low, actually, that unlikely. Now, there's multiple parts to this. It uses this 0.78 to make this calculation. Now, I went back and figured out if you can find the formula for conditional power in this. There's a beautiful normal approximation to the log hazard ratio. If you assume 0.78 and the conditional power for 16% by the end of the trial, at the time, it means that the hazard ratio was 1.03. So it was worse than 1 at the time, and the data was doing worse. The 95% credible interval for that 1.03 hazard ratio at the time of 254 events goes down to about 0.81. So somebody's calculating this conditional power for what's the chance this trial is successful, and they're using 0.78. The 95% confidence interval goes down to 0.81 as the lower bound of that. So 0.78 is not even in the 95% confidence interval of when they're making this calculation. It's a very, very unlikely value. So what is the relevance of this calculation? It's a super odd calculation. And I think it provides no information for the DSMB to make the decision. It's not a low number. You probably shouldn't be stopping trials for futility at 16%. It's not that unusual of a number. But it's calculated using, remember when we talked about the way they powered the trial, they said it has 80% power, assuming 0.78. So halfway through the trial, if you still assume that 0.78, it's gone from 80% to 16%. Doesn't seem that super odd at the time. I feel like it's not a very informative number at all. You won't be surprised to find out I'm not really a fan of conditional power. Now, conditional power assumes you know the answer and what's the probability that happens. And it uses a number that's incredibly unlikely given the data we've observed. And so it's a bad analysis in that. Now, what would I do? I would do a Bayesian predictive probability. I would use the current posterior distribution of given 254 events with a current hazard ratio of 1.03. What is the posterior distribution of the hazard ratio? And then given that estimate of the hazard ratio, what's the probability the trial is going to demonstrate superiority? Okay, I did that with this data, and it comes out to be about 1.4%. So given the current data, the and the current posterior distribution of the hazard ratio at the interim, integrating over that, the probability of seeing statistical significance at the end of the trial is 1.4%. Now, I think that's a really, really useful number. And by the way, it's a number we use. It's a calculation we use very frequently for stopping for futility because it incorporates the uncertainty at the interim, the variability of the data to come, of demonstrating superiority or the goal of the trial. It's a beautiful number. And you can use prior distributions for that are informative. You could use fairly weak priors. That analysis prior that goes into that's a word that the FDA draft guidance used. The analysis prior that goes in to calculate that could be based on an optimist's view. And when an optimist thinks it's unlikely, you could stop the trial. Now, if your optimist believes 0.78 with no variability, that's the conditional power. That's equivalent to what they calculated. You have a prior probability that the hazard ratio is 0.78 with zero variability. You think the chance of winning this trial is 16%. I don't think that optimist would stop the trial, but I think it's not the right calculation at that particular time. Okay, so a lot went into that. Now, the other component to it is that at the time they saw increased major bleeding, which was the safety endpoint, which is what anticoagulations do. You know the bleeding's going to be higher, but you're hoping it's offset by reduction in cardiovascular events. Now, the final data, I don't know what the data were at the interim on bleeding. The final data on bleeding was for the rivaroxaban arm. It was 8.8% major bleed. That was 64 events on that arm, 44 events on placebo for a 6% rate of major bleeding. So that went from 6 to 8.8, which surely went into the DSMB's decision to recommend stopping the trial. The probability of showing superiority, and I think they used an awkward number for that, but with this elevated rate, now we know major bleeding is going to be higher. That's what the drug does. But the perception of this 2.8% increase with a hazard ratio of 1.3 at the time had them recommend stopping for futility. Now, the final result of the trial was a hazard ratio on cardiovascular events that was 1.09. There was additional follow-up. This July event, they stopped the trial a month later, so there were deliberations on this, but they stopped it. There was additional follow-up, probably additional events going through adjudication. With the final events, it ended with a hazard ratio of 1.09 in that. 13 events per 100 years on rivaroxaban, 11.8 on placebo. It was doing worse than placebo at this particular time, and stopped with 1,463 patients randomized, about 400 and some plus patients less than what they planned to enroll in it. Okay, so overall, what does this mean? So I think the DSMB got it right. Knowing what I know of the results, I would have recommended stopping. But I think the problem is, I don't think that's the group to make this decision. The thing that bothers me here is there were no prospective futility rules. So now you turn over this trial. It is an incredible amount of effort by a ton of people, 1,400 patients agreeing to enter this trial, and there's no rules for stopping the trial for futility. You leave it up to smart people on the DSMB. But those are really hard decisions for a DSMB. I think this is something that the PIs in the trial should define. They should set up what is futility? What is should it be a balance of bleeding to that? Are they happy that the trial was stopped at this particular time? It took about a month before they eventually stopped it. Did they review the data? Did they agree with this? I feel like it has to be part of the design. It's frustrating as a statistician to read trials like this that don't have futility rules set up. It's a 5-year trial with 1,900 patients. Cardiovascular death is part of this endpoint, major bleeding. So I think the DSMB did right, but they're in a really hard spot to make a decision for futility. Is 16% right? Is that conditional power number the right thing in it? Now, this is a trial looking at a readily available treatment. So this is kind of comparative effectiveness, if you will. I don't think this is going to result. Maybe it results in some level of guideline saying, oh, patients with CKD stage 4 or 5 should take low-dose rivaroxaban if the data were in a particular situation. But it's not regulatory approval. It's not a sponsor going after this. I don't believe in the setting. But especially in sponsor-designed trials, the sponsor should make the decision about futility. When is enough? When should we stop the trial? It's a business decision. It's a money decision. There's an ethical component to it about asking patients to be randomized into a trial with a very low likelihood of demonstrating a result that changes practice. And so are they contributing to science if the probability of success is 1.44%? All these questions should go into the design of the trial, not humans that have to sit around and make this decision post hoc in it. The last part about it is I think conditional power is just a bad tool for that. It's a bad tool. Integrate over the uncertainty of what you know at the time. Reflective, that 0.78 was very, very unlikely. So 16% is just the wrong measure. Whether it's big or small, it's the wrong measure. So I'd love to see them use a much more realistic, relevant calculation in that. Okay, so that was article number 1 that shows up in my email box. Article number 2 is the Victory randomized clinical trial. Love the names. And this one caught my attention right off the bat. This is the first one I looked at. This came out June 10th, published online in JAMA, June 10th, 2026. And this is the Victory trial, and it is high-dose intravenous vitamin C, studying mortality and organ dysfunction in severe burn injury. I don't know much. We've done a little bit in burn, but I don't know much about it. The syndrome within it, you can imagine the clinical syndrome of this, but I don't know much about treatment in this or endpoints. So, but it struck me was the vitamin C part of it. Why did vitamin C strike me? Well, in our REMAP-CAP trial, we investigated REMAP-CAP and LOVE-IT. LOVE-IT was a trial investigating vitamin C in sepsis at the time, and COVID, the pandemic, broke out. The two trials combined their data together to investigate and randomize patients with COVID and investigating, does high-dose vitamin C improve outcomes in severe COVID? And our trial came out with, and the endpoint was organ support-free days. And in the back of my mind, I remember this result. It came out, it estimated essentially harm on the ordinal endpoint of mortality and then organ support-free days. It came out with an adjusted odds ratio of 0.88 less than 1 is harm, with a 91.4% probability of harm. It was doing worse. On mortality, so on survival was 57% in the vitamin C group and 60% on the non-vitamin C, the control. So it did worse. The trial was stopped for futility in that case. So I knew this vitamin C did not do well in COVID. And then I saw that Lovett read out in sepsis. And so the paper came out in 2022, reporting the results of vitamin C compared to placebo in patients with sepsis. And the primary endpoint in that trial was persistent organ dysfunction or mortality. So they died, or I believe at day 28, they still had persistent organ dysfunction. They couldn't get off organ support, a bad outcome. It was a dichotomous outcome of that. And in the vitamin C group, the proportion of patients that met that condition, died or organ dysfunction, was 44.5%. In placebo, it was 38.5%. So a 6% increase in that primary outcome in sepsis, statistically significant harm in that trial. So it sort of, I remembered vitamin C was not doing well in those two trials. So here, vitamin C is being given to patients with severe burn injury. So I said, I've got to see this. Does vitamin C work here? So the trial design, 1-to-1 randomized high-dose vitamin C given by IV versus placebo. The primary outcome was a composite of 28-day mortality, so clearly severe burn, and persistent organ dysfunction, defined as dependence on mechanical ventilation, kidney replacement, or needing vasopressors inotrope. So still needing, low blood pressure vasopressors. You have that support at 28 days. So this composite outcome of bad things at day 28 is the primary endpoint, similar to the Lovett trial, actually, for the burn trial. So the total sample size was planned 333 patients per group. So 666 patient trial, which would provide 78% power for demonstrating a decrease in that outcome at 27% for placebo to 18%. So that reduction, it would be 78% powered. So still relatively rare, 27% on placebo was thought to be the rate of this outcome. My first thought as a statistician, I'm just jumping in as when I read this, is, boy, there's got to be a better endpoint here. A dichotomous outcome of these bad events, but there's got to be more to this, to look at this outcome where you learn something from the 73% of patients that aren't dead or aren't in this really bad state. Now, death is worse than being on vasopressors on day 28 and likewise. So I don't like the endpoint. I'd like to see something better. Now, that isn't so much the point of this, but as a statistician reads through this, I cringe a little bit at that. Now it says, while allowing for 2 interim analyses for futility/safety. And it says the operating characteristics of the trial under the pre-specified futility rule. So they have a pre-specified futility rule here in this vitamin C may be triggered by the poor results of these other trials, the harm, the reasonable likelihood of harm. In one case, 92%, the other one was statistically significantly harmful. So the pre-specified futility rule was an adjusted risk ratio of greater than 1.1. So less than 1 is good, favors vitamin C. So at these 2 pre-specified interims, if they saw a relative risk above 1.1 favoring control, they would recommend futility. That was the pre-specified rule. And these interims were to take place at 1/3 and 2/3 of the events, that if it was above 1.1, they would stop for futility. And it says it was determined through simulation. So in the SAP, they actually go through and they look at the operating characteristics of this futility rule. And they talk about it reduces power from 79 to 78%. Futility rules reduce power because every once in a while the trial hits those futility rules, depending on what it is, and that would have gone on to be successful. Depending on that rule, you can see reduction in power. So they report what it is, and it says minimal effect on bias, which futility rules, any stopping rule has bias. Bias is not bad. And in this case, this bias is not bad. So they say it's minimal, so that's good. But they investigated, they created a pre-specified futility rule in this trial, as opposed to the track trial where there were no futility rules. Now, it doesn't mention anything about efficacy. So it looks like they did 2 futility rules and not for efficacy. Fantastic in the trial. Now, I might throw out a little bit of superiority at the time, but maybe they wanted enough data to demonstrate and change practice in that. Okay. So 2 interim analyses were pre-planned at 222 patients, 444 patients of the 666 total sample size in it. By the way, I wonder, again, statistician reading this, they report the sample size in the trial as 333 per group. And then later they talk about the interims happening at 222 total and 444 total. Did they not want to write the sample size of 666? I throw that out there as a possibility. So they said it was 333 per group, is 1.1. And so what happened in the trial? This is where they report that. This threshold was crossed at the first interim analysis, prompting the DSMB to recommend termination of the trial. By the way, I think that's a very reasonable thing to do, that you have pre-specified rules, but you have a human group that's looking at the data and they say, yes, we think it's appropriate. But you've predefined what that is in the setting. So predetermined rules hit the trigger. DSMB said, yes, you should stop the trial. And the trial was stopped in this setting. Okay. What were the data? The data were on this endpoint where it was at 120 patients randomized on vitamin C, 41%. Remember, they talked about making 27% on control go to 18. There were, I'll do placebo first. 30% of placebo patients met this endpoint, very close to the 27 that they said, which was a pretty good design estimate of that. It was 41% on vitamin C, an increase from 30 to 41%. The Fisher exact test p-value at the time was 0.08, of course, on the way to harm in that setting. I don't know why you give 2-sided p-values in a superiority trial, but we can interpret that Fisher exact p-value, of course, going the wrong way in the setting. The mortality rate on placebo was 8%, so 9 of 118. It was 15%, 18 out of 120 on, I'm sorry, I may have said that wrong. On placebo, it was 8%, 9 of 118. On vitamin C, it was 15%. So it went from 8 to 15%, almost a doubling of the mortality rate within the setting. and it hit the relative risk. The final relative risk was 1.38, which hit the 1.1 and the recommendation to stop for futility. Okay, so overall within this, and by the way, the result, I love reading the conclusion. So I imagine clinicians don't have time to read all these results, but they want to get the highlights of this. And that's why I think this little cartoon they do in JAMA, and I think New England and other journals do that, is a summary of the trial results. And many times I disagree with the conclusion. I strikingly disagree with it. I think it's bad. But here it says the conclusion is high dose intravenous vitamin C did not reduce mortality or organ dysfunction. So again, it didn't reach statistical significance and may be associated with harm in patients with severe burn injury. I think that's pretty reasonable in this setting. It wasn't statistically significantly harmful. This p-value, if we do a Bayesian interpretation of the p-value, there's a 96% chance of harm. So I would have been upset had they just said did not reduce mortality or organ dysfunction. I think there's reasonable evidence that it is harmful. And of course, in my results with vitamin C seems to be harmful for people with severe disease. This is a third result now where it does worse and seems to be harmful. So it says may be associated with harm. I thought very reasonable conclusion. Okay, so I liked this. I liked this that they put this futility rule in there. By the way, I think it could have stopped earlier than that. In the trial. I think had they seen it even earlier, they might have stopped for futility, but they had it set up. Now, I don't like, and by the way, it stopped at 33% of the sample size. So it stopped at roughly 240-ish patients rather than 666 patients, and likely doing harm to those patients, certainly not contributing to scientific results that are going to change care in this setting. Of that vitamin C is a good thing to add to the way to care for patients with severe burn. I don't love the rule of relative risk greater than 1.1. At a third of the way through the trial, 1.1 is a very different predictive probability than 2/3 of the way trial, a 1.1. And now this isn't where the number of events. This could have been an event-driven trial, but it's rarely done in sort of a binary yes-no at day 28 kind of thing. So they do number of patients. So had the events gone up and down, 1.1 is a very different statistical conclusion as to the likelihood of success of the trial if the event on control would have been 15% or 40%. It means very, very different things a third of the way through. Now, they simulated the rule and presumably under a number of different scenarios, different control rates, but I'd much prefer predictive probability in that. I think I'd even prefer conditional power, but using the MLE, I don't love it, and there's better ways to do it, but then just an absolute value. But when you do sufficient simulations of that and you time it and carry out the analyses at the appropriate time, that's a reasonable summary of the data. Now here, there could have been less events and it's a different kind of thing. So I don't love the rule, but I love the fact that they did futility, was pre-specified. They carried out the futility analyses, kind of a no-brainer that this trial, very low likelihood of success and high likelihood of harm. By the way, I should have calculated the Bayesian predictive probability at the time, much less than 1%, no chance of success in this trial. On it. So I commend the designers on it. I would have done it differently. Lots of flavors to that. So 2 papers in JAMA. Interestingly, a lot of people talk about these medical journals as, they only publish positive results, publication bias in the setting. Here are two papers published where futility was the result of both trials, and they got published in JAMA. I think really important results, I suspect. I don't know whether vitamin C is commonly done in burns in that, but I imagine anticoagulation is used and not that uncommon in patients with chronic kidney disease in that. So very, very important results in it. All right. I leave you with, when you're designing trials, design futility. It's a really important part of the trial design. Don't leave it to the DSMB to make hard decisions in this. Help make those decisions ahead of time. I appreciate you joining me here. I'll look at several papers. Again, we can go back to the design and think about them from the results. But I appreciate you joining me here and joining again. And until next time, we will be here in the interim.